that's the alpha carbon. So you attack the carbonyl, and you end up making something that looks like that, sort of. Okay? Actually, it looks like this if you want to get really technical. Okay. Now, that's what happens with number 1. What's happened with number 2 is nothing. So now you've got number 2 here as the guy on the end. And it's still connected to number 3, and so forth and so on. Okay? So you've chopped off the first one. Now, if you want to get really technical, this sucker rearranges. Okay? Because that's not stable. And you end up doing this. But the point is, what you've done with number 1 is you chopped it off. I'm not going to require you to draw the structure of that before anyone raises their hand. Okay. All right. The important thing for right now is you chop number 1 off. Okay? And you create this, and we can isolate this guy. What we do next is, after we've done the reaction, after we reacted with anhydrous HCl, okay, the next thing we do is we throw in the solvent like dichloromethane. We throw in dichloromethane to collect this guy. The rest of the protein chain is not going to be soluble in dichloromethane, because dichloromethane is a very nonpolar solvent. Okay? So it collects this thingy here, and the rest of the protein is left intact. Okay? So we can take this guy off to a chromatography system and identify what it is. Okay, now, that gives us number 1. I'd already given you a way of figuring out what number 1 was. But the difference is we haven't done anything to the rest of the protein. And now our new end guy, the new thing on the end, is number 2. So what the heck? You say we do it again? You want to do it again? So we do the whole thing again. And that identifies number 2. And that leaves number 3 as the end. So we do it again. So we sequentially chop off all the amino acids, one by one, and we turn them into this weird derivative thing, and we identify what they are. So we chop them off one at a time and identify them. And that is how we sequence a protein. Now, if you're thinking, God, I hate to sit around in a lab all day pouring a little of this in and then squirting a little of that in and then a little of this. You'd hate to do that, right? The beauty is this could all be automated. You can have a machine that does all this. You put the protein in the top of the machine. First you squirt in some of this stuff. Then you squirt in some HCl gas. Then you squirt in some dichloromethane to free out the first guy. Then you squirt in some more of that. Then you squirt in some more HCl. Then you squirt in some more of this. Collect number 2, and you just keep doing this. And the companies will tell you that you could go all the way up to maybe 200 amino acids. They lie, they exaggerate. You know, it's sort of like British Petroleum telling you that they do save oil and oil. They lie. Okay. But you can practically do 100 or maybe 50 to 100 amino acids. So you can get a good part of a protein. Okay? This way. In one afternoon of collecting. Now, you're saying, but my protein's a lot bigger than that. You know, a protein chain could be 200, 400 amino acids. I want the whole thing. So here's what we're going to do for you. Here's what we're going to do. We're going to take that big, long protein, and we're going to chop it into manageable chunks. So we're going to take a protein that's got, say, 200 amino acids. And there are various proteins, or enzymes around, that digest or break apart proteins. Just like I talked about restriction enzymes, recognize particular DNA sequences for cleavage. You know, there was one that recognized GCGC, there was one that recognizes GATC. You know, there were enzymes that recognize particular DNA sequences. There are enzymes that recognize particular patterns of amino acids. You've heard of these things. Your body has them. You use them to digest the proteins in the food you eat. Okay? There are things like trypsin. You've heard of trypsin, right? Chymotrypsin. Pepsin. Papain. There's a whole bunch of these guys. And they all recognize somewhat different sets of amino acids for cleavage. Okay? I'm not going to make you all learn what they all recognize. If you really want to, go ahead. But, for our purpose, just knowing that there's a collection of proteins that work, or enzymes that work sort of like the nucleases work with DNA. And what we can do is we throw one of these guys in with this protein, and it's not going to chop it completely down to the down to the amino acids. It's going to chop it apart in chunks. Like trypsin might cut here, and here, and here. And so what we'd end up with is making a series of fragments. And I already told you how we could isolate the fragments. How would we isolate them? We could do electrophoresis on them to collect those guys. And so we're not going to try to sequence this whole thing. We're going to sequence these pieces. Now, here's the catch. You knew there was a catch, right? Okay? We got these pieces, A, B, C, D. Okay? They're not going to, if we run our gel, or for that matter, if we ran a column, because we could also do this with a column, they're not going to come off necessarily A, B, C, D, are they? Right? How are they going to come off? Based upon size. So you might end up with A here, and B here, and D here, and C there, depending upon the size. Okay? So we're going to isolate these pieces. We're going to isolate these four pieces, and we're going to sequence them, but we don't know how to put them together. Because we don't know, just based upon this, there's no little spray paint tag on these suckers saying that this one's A, and this one's D, and this one's B. Okay? There's no label on these guys. There's no little man, little elf spray painting them. Okay? So, here's how we get around that problem. Here's how we get around that problem. We take our whole protein here, and we cut it with one enzyme. And we've got these chunks. Okay? And then we take another batch of that protein, and we cut it with a different enzyme. It cuts in different places. So we pick another one of these guys. And that way, I got a chunk here, 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 and here. Okay? Now, if you look at what we've got here, okay, where this one cut, this enzyme didn't cut. So the pieces where one of them is broken, the other one's intact. And we look for the regions of overlap. Okay? All right? And that tells us what order we're going to do them in. And I have a cute little example for how this would work. Now, let's suppose I tried this thing. I'm not going to do something that's 200 long. That would take all the fun out of this. Okay? Let's say I taken a protein. And the first thing I've done is used a method where I found out what the N-terminus is. And so I tell you all, like this, think of this like the problem on your exam. I tell you that the N-terminus is H. So you know the very first one's got to be H. And with my first enzyme, okay,